So here we're going to be looking at protein structure. We've seen this initial image, different ways of representing structure to try to show the three-dimensional shape, because for proteins, as we're going to see here, shape is very important to their proper function. So just to give you a basic idea, is there's four main stages of organization. This is just the amino acid sequence, and this is going to take on an initial shape, and it's going to start to form, and ultimately here, the fourth stage of protein structure is ultimately to make a functional protein. So the general overview of protein structure levels, we have the assembly of the amino acid sequence, the initial folding, where we're forming alpha and beta sheets. We have the packaging here, where we're starting to get a three-dimensional structure, and then interaction, where we have a complex of a protein molecule making this functional protein in the end. So starting with the basics, protein assembly, the primary structure. The specific sequence of amino acids is what's going to form a protein. So we're looking here at a ribosome. This occurs in the cell, and this is cytosol. And this involves polymerization of amino acids attached to the tRNA. So here's our messenger RNA here, encoding for the sequence. We have our ribosome, and we are building our primary structure, which is our tRNAs, bringing in the amino acid sequence, binding here, allowing us to follow the messenger RNA sequence for amino acids. This is yielding the primary structure. This is just the amino acids being linked together. Now those amino acids that form a linear kind of chain here, this is only one-dimensional. A perfect linear amino acid polymer is neither functional nor energetically favorable. Folding needs to occur. So this is just the link of amino acids, but now we need folding to take on the proper shape. And what we see here is that initial folding is creating now our secondary structure. This is folding, and this shape is taken on because of hydrogen bonds. And again, this occurs in the cytosol of the cell and involves localized interactions, which is the primary structure here developing an alpha helix. This would be a beta sheet. These are both part of the secondary structure that's occurring. So that secondary structure is the chemical nature of the carboxyl and amino groups of all the amino acids. This permits the hydrogen binding to form. This is causing these molecules to become more stable. Hence, this defines the secondary structure within a protein. And as we see here, alpha helix and beta sheets. The R groups, or the variant group, is what impacts the likelihood of secondary structure formation. So primary structure, we see our amino acids listed here. Now we're looking at the secondary structure. That secondary structure is nonlinear, starting to get that three-dimensional shape. And this is because of those variant groups that are now able to interact. The interaction, as we see here and here, is that hydrogen bonding. And there's also van der Waals interactions causing this structure to take on this specific shape. As I said, there's alpha helix and beta sheets, and they have regular hydrogen bonding patterns, and we can classify them as that. The beta sheets, as we may assume here, forms a very flat structure. The alpha helix kind of looks like that DNA. We're kind of getting that wrapping that does occur. This cytosol, we're looking at now our tertiary structure. It involves interactions between secondary structure elements, yielding the next level, our tertiary structure. So you can see right here we have an alpha helix, here we have a beta sheet, now we're getting further interaction that's occurring. These tertiary structures, this final three-dimensional shape of the protein, you can see that image here, they're nonlinear and they're three-dimensional. So that's why it's important to show this rotating, so you can get appreciation for the unique three-dimensional structure that it does have. Now protein interactions, so we see here tertiary or quaternary structure. So now we're getting different interactions that occur with these polypeptides. There's multi-components now interacting. This does still occur in the cytosol, and it's proximate to other folded and packaged proteins. This involves interaction among tertiary structure elements separate into polymer chains. These are nonlinear three-dimensional structures. You can see the example here, kind of a cartoon image, but this is hemoglobin. You can see distinctive 1, 2, 3, 4 different structures here, all coming and interacting with one another to form a functional final protein structure here. So that three-dimensional shape, as we see here, very important. That's why I show it in a rotating basis. You can get an idea of the uniqueness from all different angles here. Now, each protein has a unique structure. That's important to remember. Now, that amino acid sequence, that free energy mineralization leads to that structure, and the dynamics will then lead to the proper function. So this amino acid sequence, what looks like kind of a garbled kind of misprint here, each one of these letters represents an amino acid. Those amino acids form a chain, and through the structure and ultimately the dynamics to form the proper function, that folding process will make this amino acid sequence a functional protein. Now folds can be used for classification. There's a simple fold, is architecture, which is the overall shape and orientation of the secondary structure, as I mentioned here, as alpha and beta barrels, for example. We can classify these as what we call fold families. This is a categorization that takes into account the empirical or biological properties. This is important because shape determines function. Here we're kind of classifying different shapes. We could also classify them as superfamilies, which in addition to the fold families, includes evolutionary or ancestral properties. This gets into trying to track different proteins or shapes over the course of evolutionary time. So this is an interesting video here that helps you understand the importance of protein folding. And because protein folding does occur, we need to have a way to allow proteins or amino acids to fold properly. So to help newly produced proteins fold properly, we have something called chaperone proteins. And this is what they look like. There's literally, it looks like a donut here. There's literally this kind of protective interior structure. These chaperone proteins allow a protective area to allow these amino acid sequence to fold into proper functional proteins. So if these chaperone proteins have deficiencies, they play a role in certain diseases. Cystic fibrosis and Alzheimer's disease is an example of two of them, where proteins don't fold properly. So to kind of go over that same thing, we have a primary structure, which is just the amino acid sequence. That then will lead to secondary structures, which are alpha helixes and beta sheets. Tertiary structure is the initial three-dimensional structure formed by the assembly of those structures. And lastly, the quaternary structure. This is where they're interaction forming with multiple peptide chains. It's just another way to kind of look at it. With the amino acid sequence, we're getting into initial structures. They're kind of forming a three-dimensional shape, and then there's interaction that's occurring here at the final stage.